Epigenetics. What is epigenetics? Epigenetics is the phenotypes and outside stuff that affect gene expression, leading to differences in expression of things like obesity and cancer. It is the study of heritable changes in gene expression or cellular phenotype caused by mechanisms other than the changes in underlying DNA sequence. Epigenetic marks include covalent DNA modifications and post-translational histone modifications. Why are our cells different? Gene expression has a big role in this. So, let's talk about gene expression. What is gene expression? Every cell has the same DNA code, but different genes, sections of DNA, are used for one cell type versus another. An example is liver cells versus muscle cells. DNA is coiled up in the nucleus of the cell, while genes are a section of DNA that codes for particular traits or characteristics. Gene expressions can be passed on through generations, but the genes themselves are only not modified. In other words, we have changed the phenotype without changing the genotype. There are multiple mechanisms of epigenetics, the most common being DNA methylation and histone modification. DNA methylation is when methyl groups are added to the DNA molecule, thus suppressing gene expressions. This tightens the coil of the DNA, making it harder for enzymes to access and activate a gene. DNA demethylation is the removal of a methyl group from DNA, which has the opposite effect in that it will loosen the wrapped DNA, thereby making certain genes more accessible to be activated by enzymes. So, what are some life examples of epigenetics? Agouti mice are fat yellow mice given a good diet because baby mice need to be normal because of activation or suppression of certain genes. Similar examples of this were the Dutch famine of 1944 to 1945 and the Sweden famine.